the September 13th, 2017 meeting of the Sacramento Regional Sanitation District. Would the clerk please call the roll to establish a quorum?
Okay. Good morning, Directors Ashby, Bruins, Frost, Guerra, Harris.
Here.
Hume.
Here.
Kennedy.
Here.
Lee. McGarvey.
Here.
Morin. Natoli.
Here.
Orozco.
Here.
Peters.
Here.
Cerna.
Here.
Villegas.
Here.
Warren. Carr. You have a quorum.
Thank you. Would you please join me in the Pledge of Allegiance?
I pledge allegiance to the flag of the United States of America and to the republic for which it stands, one nation, under God, indivisible, with liberty and justice for all.
This meeting of the Sacramento Regional County Sanitation and Sacramento Area Sewer Districts is being broadcast live on Metro Cable 14, the local government affairs channel on the Comcast, Consolidated Communications, and AT&T Uverse cable systems. The meeting is closed captioned for our hearing impaired viewers and webcast at www.sacmetrocable.tv. Today's meeting will replay Sunday, September 17th at 6 p.m. on Channel 14. In accordance with Government Code 54952.3, compensation for meeting of these legislative bodies is required to be verbally disclosed. The amount of $100 will be paid for each member participating today as a member of the Sacramento Regional County Sanitation District, and the amount of $100 will be paid for each board member participating today as a member of Sacramento Area Sewer District. Compensation for Sacramento County supervisors and City of Sacramento council members is paid to the county and city respectively to partially offset the cost of those governments. Compensation for other board members is delivered to the individuals. Members of the audience wishing to address the board should fill out a speaker identification form located at the back of chambers and hand it to the clerk. Please speak into the microphone when addressing the board and state your name for the record. Please silence your electronic devices at this time. Thank you.
Thank you. And will you please call the first item?
Okay. Acting as Sacramento Regional County Sanitation District, your district consent matters are items 1 through 3.
Are there any questions of staff on the consent items?
I'll move consent. Second.
Moved by Hume, seconded by Mr. Harris. All those in favor?
Aye.
Opposed? Abstain.
Madam Chair, I'm here. Sorry, I should have. There's Eric Wink, Director of Delta Protection Commission, here to speak to the board for a couple of minutes, and I don't think he filled a card yet. He just walked in, so I just wanted to remind him.
Oh, okay. At what point?
Right now.
Oh, okay. All right. Sir, you're up.
Great. Thank you, Mr. Chair. Members, I'm Eric Vink with the Delta Protection Commission. I'm just here very briefly to give a big thank you to Regional San. You all might know that this Saturday is River and Coastal Cleanup Day throughout California. This started out on the coast. It's moved its way up to the inland waterways. And the Delta Protection Commission is sponsoring 5 locations throughout the 5 Delta counties. One of those is in Yolo County along the Sacramento River near the Stone Locks, where the little arm of the Sacramento River reaches into the Stone Locks area. And we're very appreciative of Regional San for providing some sponsorship support so we could provide t-shirts to our participants on Saturday. Most of these will go to the people that are participating in the river cleanup along the Sacramento River. We'll send some of those t-shirts on to our other Delta locations as well. Especially want to call out our commission members, Supervisor Natoli and Supervisor Villegas. Supervisor Villegas is a real stalwart with this event. He provides the food, and we'll probably have between 75 and 100 people there working on cleaning up the river. So just want to thank Regional San. You all are a great partner on this. I think there's also a cleanup at the buffer lands as well that our sister agency, the Delta Conservancy, is sponsoring. But just again, thank you very much.
Thanks, George. Thank you. Any questions, comments? All right, is there any other member of the public that would like to comment? Next item.
All right, acting as Sacramento Regional County Sanitation District, item number 5, results of a scientific study examining the effects of treated wastewater on phytoplankton growth.
It's a little bit of a change up you'll get to hear a little bit of science things mixed in with all our contract awards and things. So.
Good morning, Chair and members of the board. My name is Tim Musson, and I'm a scientist in the scientific research section of the Policy and Planning Department at Regional SAN. Thank you for this opportunity today to share with you the results from a scientific experiment that was supported by Regional SAN. So one of the activities of the scientific research section is to help perform studies that evaluate potential effects from our treatment plant's discharge on the ecological health of the Sacramento River. And this study directly supported this activity by asking, does wastewater-derived ammonia affect phytoplankton growth or phytoplankton species composition in the Sacramento River? This was a collaborative study that included scientists from the United States Geological Survey, multiple local universities, and Regional SAN. And the funding for the project came from Regional SAN and also from a research grant by the Interagency Ecological Program. So to help you understand the study, I'd like to provide you with some background on phytoplankton. Phytoplankton are tiny microscopic plants that live suspended in river water, and they are food for many other aquatic organisms, such as tiny zooplankton, which in turn are eaten by fish, birds, and other wildlife. And phytoplankton biomass has been observed to decline down the length of the Sacramento River. And some researchers hypothesized that the ammonia within the discharge from the Sacramento Regional Wastewater Treatment Plant was contributing to this phytoplankton decline by reducing the phytoplankton's growth rate. So this experiment directly tested phytoplankton's response to the presence and absence of wastewater effluent in the Sacramento River. And to help you understand the methods in this study, there is a diagram for the Sacramento River located on the left side of the screen, which shows the area downstream of our treatment plant. So in this diagram, the gray bars represent regions of the river with the normal presence of diluted wastewater effluent, and then the white bars represent smaller experimental water parcels that we created that had no wastewater effluent added to the river. So the way that we conducted this experiment was by diverting the discharge into storage basins at the treatment plant for about 19 hours, which created a 6-mile-long stretch of the river in a parcel of water that was effluent-free. Then the water parcels were tracked by boats as they moved downstream, and we repeatedly sampled the nutrients and the phytoplankton over a course of 5 days to look at the changes in the community. And to help understand this project, you can envision yourself sitting on a boat that's moving downstream with the current, and then repeatedly sampling the water below you for the phytoplankton and nutrient composition. We also had a second boat located further downstream in a separate water parcel that had the regular diluted effluent present. And this way we could compare what was occurring in the two different parcels to determine the effects of the ammonia and the effluent. So the map on the right shows the sample locations for this experiment. And on it, the gray marks show the location of the water parcel that would receive wastewater effluent under normal conditions, similar normal conditions, and then the blue marks represent the water parcel that would remain effluent-free as it moved downstream. The experiments were conducted both in the spring and in the fall, which are generally high periods of phytoplankton growth for the river. And we started above the wastewater treatment plant near Land Park, and then followed the pulses as they moved downstream past the town of Ileton. These experiments were conducted during the drought, and therefore there was really unusually slow river flows during this period, and also long transport times. And while the overall flow of the river remained in a downstream direction, the tidal forcings were actually strong enough during this period to push water back upstream with reverse flows for a few hours twice a day. So to track the water parcels as they move downstream, we used two different methods. One was by employing little drifting floaters at the surface. So these had a little flag that you could see from the boat, but a very low surface profile so they wouldn't be affected by wind. However, under the water there was this large sail that would catch the river current and move the floater downstream or upstream at the same time that the water was moving in the parcel. Then the boat would try to remain near the floaters. The other technique that we used was high-frequency water meters, and these would be measuring the water quality every second within the river. So therefore, you could tell if the boat had moved out of the water parcel because the meter would detect slight changes in electrical conductivity and other water quality parameters to show that you've moved from one patch of water to another. We measured a lot of different water quality variables during this experiment, including the nutrients ammonia, nitrate, and phosphate. And then we also collected many biological measures, and that's the list on the other side of the screen. And one of those was chlorophyll A, or the total biomass of the phytoplankton that were present in the river. Another was phytoplankton enumerations, and this allowed us to identify the individual species of phytoplankton that were contained within the water samples. So the main question from the project was, how did the ammonia and phytoplankton concentrations differ amongst the two water parcels as they moved downstream? So at the bottom of the figure, you can see the travel time during the 5 days of the experiment, and time 0 is marked at the point when the water parcels passed the discharge point for the treatment plant. So when we look at the upstream ammonia concentrations, we see that in both water parcels they were quite low. Then, after moving past the treatment plant, the water that received the wastewater effluent jumped up to higher ammonia concentrations, and the water that did not receive wastewater effluent remained low in the downstream section of the river. When we look at how the phytoplankton responded on the next figure, still time 0 is the point when the parcel had passed by the treatment plant. And in the upstream area, we see that there was a large decline in phytoplankton biomass occurring the day before we reached the treatment plant. And then, going downstream, the water parcel that received effluent continued to decline, but at the same time, the water parcel that was effluent-free showed a very similar decline in chlorophyll concentrations. And this leads us to believe that it was not the ammonia within wastewater effluent that was driving this decline downstream of the treatment plant. Another question from the project was, can desirable species of phytoplankton grow in the river water when the wastewater effluent is present? So when you look at the ordination figures that are shown here for June and October, they show the distribution of phytoplankton enumeration samples. And it has a very similar distribution between both parcels. And that means that the phytoplankton species that were present in the river were basically the same in the presence or absence of wastewater effluent. And when we look at what species of phytoplankton were living in the river at the time, 90% of them were diatoms. And these are considered a very desirable food source for other animals within the river. So you'll recall that there was a large decrease in chlorophyll A concentrations, and we wanted to test if perhaps another factor was contributing to this decline. And that would be, were the populations of grazing organisms at such high concentrations that they could eat the phytoplankton faster than they could grow. So I've put a simplified delta food web here on this slide, and it shows how phytoplankton take up nutrients to support their growth, and then zooplankton and clams eat the phytoplankton. And then small fishes such as delta smelt might eat the zooplankton to support their population. So we took nets and we went out and we collected the zooplankton from the river, and we saw that their biomass was not high enough to be causing this phytoplankton decline. We also took a trawl behind the boat and scraped clams up off the bottom of the river, and we saw that the clam biomass as well throughout most regions of the river really wasn't substantial enough to be driving the phytoplankton decline. Therefore, the study concludes that there's other factors that are probably at work here. There might be an overall lack of sufficient light to sustain phytoplankton growth in the river. And also, the changes in the river hydrology, or the movement of flow through the river, might allow the phytoplankton to be settling down to the bottom and become stuck in the sediment, so they can't keep moving downstream and growing. So overall, in conclusion from this experiment, the main finding was that effluent-derived ammonia did not cause the decline in phytoplankton biomass that was present in the Sacramento River, and it also did not change the species composition of the phytoplankton that were growing in the river. The phytoplankton species that we did observe were a desirable, high-quality food source for other delta organisms and organisms in the Sacramento River. Also, the phytoplankton decline was not being driven by grazing animals that were consuming the phytoplankton as they moved downstream. And overall, future research should probably investigate other factors, or include other factors in the investigations, such as light availability or changes in flow patterns. Thank you for your time in hearing this presentation. I'd be happy to answer any questions you might have.
And we have some. Director Cerna.
Thank you, Mr. Chair. Thank you for the information. This is very interesting. At least I find it pretty fascinating. A couple of questions. The first is, do you expect to, or perhaps you already have, given a presentation to the Regional Water Quality Control Board about this?
Right, so we haven't directly gone to the Regional Water Quality Board. We've presented the findings at a number of conferences which they attended for the whole Delta scientific community. But I haven't heard plans yet to directly discuss it with them. They now have a nutrient specialist who is working on a nutrient work plan for the Delta and system, and we are involved in many work groups with their staff, and they're aware of the findings from this study and are incorporating them into the work group and design for the future.
Okay. I think it's important that they understand some of the conclusions from the study, and then lastly, I guess how important is the footnote to this, that the field study was done during a time of drought? In other words, would we expect that there would be drastically different conclusions if we had a normal amount of flow or an excessive amount of flow?
Right, so during the drought, you could almost consider this the most likely period to have the effluent affecting the phytoplankton.
So it's extremely conservative.
Right, so during this time, there was a longer exposure time that we were able to track down the river with long travel times, and also the concentrations are higher than average during the drought years because we had less dilution in the receiving water for the phytoplankton, or the nutrients.
Thank you.
Thank you.
Director Harris.
Thank you, Chair. Actually, Phil touched on one of my questions, being the effects of the drought on the study, but one factor I didn't see that you took into effect is temperature. So
Right.
the temperature of the water certainly would have been higher in a low flow condition. I would think that that would affect phytoplankton significantly. So was that one of the control factors that you observed?
No, you're absolutely correct. We did have higher temperatures during the experiment than what you would see during a higher flow year with colder water moving through the river. And higher temperatures can allow phytoplankton to grow at faster rates under ideal conditions, but it can also allow the organisms that consume them to be growing at faster conditions. So you generally would expect a quicker response during a warm period. We don't have slow enough flows to get stratification occurring within the river. Even during the drought, the water was well mixed. But if that were to occur, such that the surface water stayed up near the top and layers form of thermal inclines, then you could really see a strong growth of phytoplankton that might occur. And in little isolated pockets down in the delta, that may happen.
Okay. Director Natoli.
Thank you, Mr. Chair. Yeah, I appreciate you sharing this with us this morning. I guess I have a couple questions kind of along the same veins as Directors Harris and Cerna. What about submitting this to the Independent Science Board for the Delta Stewardship Council? And I know that over the years, meetings with EPA representatives and certainly hearing from the water contractors, they've really pointed at ammonia as being a degrading factor, and they've pointed at regional sanitation's outflows, and have really made their case. And obviously it got reflected, at least in part, I think, in some of the permit requirements and the billions of dollars investments that we're making to remove ammonia from the waste stream. But I guess my question is that what about peer review? What about submitting this to, I heard what you said earlier in response to Supervisor Cerna, but I think it's important because, again, there have been others who have done studies in your same discipline who have, pointed directly at regional sanitation and made us the demon that's, causing destruction and degradation of the Delta. And I've certainly, at least from my little understanding of the science, have countered that in conversations, but certainly in other forums. And so I think it's important that if we have information that provides additional supplemental information and or contrary, and again, science obviously evolves over time, but I would hope we'd build off of this and at least provide some additional data to folks for their consideration and within the scientific community, but obviously the politics of water, people take things and run with it, whether it's based in science or not. So I'm curious about additional reviews and science panel with Delta Stewardship Council in particular.
Right. So this project, we completely agree that we need to make the information known to the researchers, policy managers as broadly as possible. And this study has been published in the peer-reviewed literature in the journal Limnology and Oceanography, and we paid for it to be open access so that anyone can link to the journal and pull out the published article for findings. So now it is within the best available science, as this is one of the premier journals for phytoplankton research. Another interesting point from this project was that some of the researchers who had pointed out the original theories are actually co-authors on this report with us and were present during the experiment and agree with the conclusions.
So they've changed their view based upon better and newer information?
For the Sacramento River. Yeah. When it comes down to the lower regions of the Delta, they feel their theories may still hold, but this study in the area that it covered, they feel pretty succinctly shows that other factors are likely to be causing phytoplankton decline within the river. And we definitely also want to promote this sort of study to management agencies as a method for looking at how these theories, hold up under actual experiments under natural systems, as they are a cheaper alternative than implementing policies to see how effective they'll be.
So let me ask you just about the way you conducted your experiment too. Had previous authors of either papers and or scientific data, had they done as thorough and had they used a similar regime to you, to floating down the river and taking parcels of water and tracking them as you've outlined?
Right, so it's really an expensive study to spend 5 days with one parcel of water as it moves downstream, and you get to cover a much smaller area. So all of the previous studies that had looked at the effects of nutrients on phytoplankton had simply started at one end of the river and moved at a rapid speed with the boat going through multiple parcels. And in that way, you can't tell if it was something from upstream that had changed the concentration and drifted through versus the actual presence of effluent in the water. And therefore, this Lagrangian style that tracked both the presence and absence of effluent was a novel experiment for the system.
And so I guess just looking forward, where do you see this body of information affecting policy? Because again, I recall a meeting in Washington, D.C. a couple of years ago with the Delta County Coalition and folks from the EPA. They were pretty adamant about the effects of ammonia, and certainly they, and I've heard it from elected representatives at very high levels that, Sacramento regional sand and, their discharge and, concentrations of ammonia and killing fish and, stifling the, food chain and so forth. And so I guess how is it that, again, with, recognizing that other research could follow on to this, but I think this being the best available science, I think it's important that that we can, and again, maybe they say because you work for Regional Sand that somehow that affects the science. So then do your own Lagrangian study and do a validation if you don't trust what we've done. So I guess I'm curious. So what happens in the scientific community, maybe Prabhakar, what happens in the realm of permitting in other agencies?
I'll jump in and address that part of it a little bit. First of all, it's tempting for us to say, well, have we spent are we spending the $1 billion that unnecessarily? The answer that is no. Because the ammonia we're asked to reduce ammonia for a number of reasons. There's new reasons since our permit was issued that came on board. For example, mussel life, where the EPA established a much lower level for mussel criteria, which would trigger ammonia removal. And as Tim pointed out, the thing that we need to remember is, while this may not be affecting in the Sacramento River, the delta still, there's a belief that the ammonia, especially where the Sassoon Marsh and where that this saline water mixes with the cleaner water from here, some scientific community still believes ammonia is an issue, and Regional Sand is largest contributor to the ammonia, especially all the way to Sassoon Marsh. So we don't need to second guess whether we're doing the right thing or not on the project-wise. The second thing is there's still a lot of focus on ammonia, not only just from our perspectives, but they're working with the other treatment plants down in the bay that they're still discharging to that water body. So specifically for us, Delta Science program essentially established $1 million study to study the effects on the river in the presence and the absence of our discharge. Remember that we are essentially converting our treatment process. They want to learn ahead of time what's happening and also what would happen when we're done with removing the ammonia. There's some concern in some scientific community whether we are removing the nutrients too quickly and too much, but that's not really at a vocal level to point that, maybe it's a mistake that we made Regional Sand to remove the ammonia out. I only jokingly say, well, give us $1 billion, we'll put the ammonia back if you want it. But those are studies. There's still focus is happening. And the better part is, like what Tim said, we are working with what were once the adversaries, and they're part of this program. They're also contributing, so that we're essentially developing this science more as together, which is important. And there's more work to be done, but this is just a very important finding that we want to share with you.
Okay, thanks. Director Vegas.
Thank you, Mr. Chair. So first of all, thank you, Tim. This is although it seems a bit wonky, it's incredibly important as we go forward, because we're probably already entering sort of the time in which we need to prepare for the next round of NPDES permits that we were challenged on how many years ago, and that was a very vigorous fight. We were accused of a lot of different things, and consequently, it cost our ratepayers a lot of money to address those reasonably, as we were trying to be considerate and good stewards of our watershed. I would suggest that we follow up with Director Cerna's recommendation of the Regional Water Quality Control Board and at least share with them the findings and offer to present to them, because I think it's important. They're a key player in our regional water systems, and I think to the extent that we've now drawn some conclusions, we ought to share those with the players that are influential in our process. And then a question. So in terms of the title reversals, I know that's a conversation that we maybe don't directly have influence over, but ultimately the reversals that you referenced will affect our capacity to continue to discharge into the river in the event that there's less water during certain times of the year, in the event that there's a conveyance system that's constructed ultimately. So how far up did you notice those tidal reversals? I mean, I know this is probably common knowledge and it wasn't specific to the study that you did, but I'm curious about that because that's going to have significant impacts going forward for a lot of reasons, not the least of which is discharging into the river with our effluent.
Right. So the influence of the tides reaches further upstream than the area with the reversal, right? So there are some regions that are quite far upstream, say near the I-80 bridge, where you'll see a reduction in velocity, but not necessarily, a reverse tide. As we get closer downstream, there will be periods where you have just sort of a cessation of flow. But for the region that's reversing, it's mostly the lower section of where we were studying. There are reversals at Freeport where we discharge that were happening twice a day during this period, and I think that it would stretch another 10 to 15 miles upstream, but that doesn't mean that the water from our discharge traveled that far upstream, right? It's just water that was located further upstream would be reversing in that place. Only less than a mile, I would say, of reversal from where we discharge.
Yeah. Well, I think that the information is timely as we sort of begin to sort of put together our application for the next round of our permitting, the application itself. So very timely. Thank you.
And in your question, you alluded to the water fix, sort of in what's happening. That is our contention, because that hasn't been properly studied. The effect on our ability to discharge into the river under the water fix scenarios hasn't been studied. That's the crux of our lawsuit. And we want to know, and we want that to be mitigated before that project ever happens. So.
Right. Thank you. Director Hume.
Thank you, Chair. Tim, thank you for your presentation. Very thorough, obviously backed up with scientific data, and presented some pretty compelling conclusions with respect to what is and is not happening. Question for you on the slide that showed the study area where wastewater present, wastewater not present. I apologize. At this scale, I can't discern the colors of the triangles and the circles, but it looks like, are there some where there was wastewater present above our outflow location?
Right, so that's tracking the locations of the parcel of water that would eventually receive the wastewater. So we started upstream of the treatment plant so we would know what conditions look like prior to our discharge. So that's where the parcels were tracked that were effluent-free, or with wastewater upstream, and then you can also see the green dots downstream. So there was no wastewater upstream of our treatment plant during this experiment. Okay. But when you're trying to identify what samples were, with the parcel of water we were following, those started upstream of the discharge location.
Oh, okay, that's what I was trying to determine. I couldn't tell if they were shaded upstream, which would suggest to me that there was effluent before it even reached our outflow.
Right, it's just labeling that one parcel of water that we tracked the whole way.
Got you. And so then my second question related to that you're tracking one parcel of water, is obviously in that float period, there's a pretty significant decline, as you mentioned, right. Particularly in the October samplings prior to hitting our outflow spot.
Right.
Are you able to identify what section of river that decline is, where that's happening?
Yeah, so we've gone and looked now at a more detailed location where this was just a few samples collected during the day. So, we could see that they were getting lower over time in that region. But I don't believe it's a dramatic, here it is and then it's gone. It's declining through the stretch upstream. And when we look further upstream in a more recent project, we saw that decline continues in further reaches as well, where it can. So I think that the fact there's going to probably be multiple factors that are contributing to this decline, and more research will probably help us locate which is having the most influence at different locations.
Okay. And then my final questions for you, Prabhakar, is the district has invested a little over $400,000 for this information. So beyond just a greater understanding and maybe some empirical knowledge, to the point brought up by some of the other directors, what tangible benefit are we going to get from having this knowledge in our possession?
The most important benefit is, again, like I said, ammonia is still looked at for other reasons. As much money as we're spending to remove ammonia, we're not able to remove 100%. There's a percentage that still will remain, number one, the ammonia, a smaller percentage. There's another form, the nitrate. There's a bigger percentage remains in the river as a nitrate form. So we want to continue to do this research because there are tightening happening. So if that results in additional treatment that we have to put in the future, we want to be ahead of that to develop good science. So our continued engagement in this is very important. We just can't let our guard down thinking that, we're doing the ammonia and then we no longer want to actually study it. That's quite contrary to that. We want to look the next levels and where it's coming down the road. The other thing is one of our values is environmental stewardship. We want to understand what's our effect is on the river. And we want to study these things and be proactive in taking care of them. And this is also another effort that fits in. So that's why this is valuable research and continue to do it.
Thank you. And you actually touched on what was going to be my follow-up question is, obviously the results of this study show a diminished effect that we're causing, at least in what the exploration was herein. But you say that we're going to continue with the studies. Do you have an idea of how much more we're going to put towards those future studies?
And not exactly this type. There are other types of studies that we would be working on. I think if you remember when a year ago or two that we brought forth, probably around $500,000 a year range is where we would be involved in terms of conducting studies, working with other universities and the programs in the range. But again, it may be more given if there's a specific need that comes in. But all for the purpose of developing the right science. If you remember, back prior to 2010, we don't have any of this information. We were very essentially caught without the right kind of information being available, just one side providing it. And we don't ever want to be in that position. We want to develop the right type of information. And again, not just for defending our permit, but doing the right thing. Mm-hmm. What the regional SAN needs to do, so that's equally important. And the sciences is very important for that.
Thank you very much.
All right, any other questions? No, thank you, Doctor. If every scientist could be as succinct and interesting, I may have gotten a better GPA in college.
Thank you all for your time.
Thank you. Next item.
All right. Acting as Sacramento Regional County Sanitation District and Sacramento Area Sewer District, item number 6 is your district consent matter.
Are there any questions, comments? Is there a motion?
So moved. So moved.
Item consent?
We're on 6.
Oh,
All right, everybody's. I'm sorry, Director Frost, what did you say?
we're on item 6.
Yes.
6.
I'll move the item.
Thank
Second.
you. We have a motion and a second. All those in favor?
Aye.
Opposed? Abstain?
I'm sorry, was that Bruins that second?
Yes.
Thank you. Okay. So I can use both of those motions, or that motion for both Regional San and Sanitation District.
Right.
Okay. Item number 7, acting as Sacramento Regional County Sanitation District and Sacramento Area Sewer District, miscellaneous director and district engineer matters.
A couple of things. First of all, we do not have a second meeting in September 27th. I already see a reaction from Director Peters. It's a happy. We also have employee appreciations happening. Actually, one's happening for the SASD staff this afternoon, and we'll have one for early October for Regional SAN. We do this annually, and to appreciate the hard work. I just wanted to report to your board. Also, if you remember, late last year, we developed strategic plans for both districts, and we have assembled a team of about 80 people within the organization to follow up on the strategic plan activities, develop the activities at more granular level. What would it take to do to accomplish our goals that we've outlined in those plans? So our goal for this is by the end of this year, we will have a list of activities for both districts for us to be embarking on in case of Regional SAN over the next 10 years, and in case of SASD, the next 5 years, to accomplish our goals laid out. So that's the effort that's happening. It's an important effort happening. Last but not least, Regional SAN, it should have been SASD as well. Those of you that are part of the county board supervisors, that you awarded an award to Regional SAN on behalf of Burke. So I'm going to let Christoph just briefly mention what that award is for.
Sure. So yesterday, we received an award, Regional SAN, and as Prabhakar mentioned, SASD as well, from BERC. That's the Business Environmental Resource Center, and the County Board of Supervisors. And it's the Sustainable Business Awards, and so these are focused on basically sustainability efforts on the part of local businesses. And so there were 6 other businesses. We were the only public agency that was there. And they were recognizing things like energy conservation, water conservation, solid waste reduction, things like that. Our award was for cultural sustainability. So it was a little different twist on it. And basically it looks at more of the people side of the organization. We do some good large environmental projects that everyone can see, but this was focused on an effort that we did with our staff. It's called the Going Green program, and it's just a variety of different ways to get staff an opportunity to hear about environmental things so that they can take that back in their personal lives. So it's it's not a sales pitch or anything. It's just information that's available. We've got some bulletin boards up. We have monthly speakers that come in from time to time at lunchtime. There's going to be an e-recycling or e-waste recycling effort in a couple of our locations. So just things like that, and that's what we're recognized for.
Great. Any questions? Is that it, Prabhakar?
That's it. So, thank you.
Next item.
Okay. Acting as Sacramento Area Sewer District, your district consent matters are items 8 and 9.
And we have people punching in. Director Frost.
I need to pull item 8 and recuse myself due to a contribution received within the last 12 months for item 8.
So you're recusing yourself or you're pulling it?
Well, I need to recuse myself. I don't know if I need to walk
just
away.
Just recuse yourself from it there.
Okay.
And if there's discussion, then excuse
All right.
yourself.
Move the
Director
items.
Yeah, Mr. Chair, out of abundance of caution, I too am going to recuse myself on item 8.
Okay.
Second the motion.
Who moved? All right, Director Natoli moves consent. Director Peters seconds with the notation that Directors Cerna and Frost have recused themselves from item 8. All those in favor?
Aye.
Opposed? Abstain. Next item.
Acting as Sacramento Area Sewer District, item number 10, adopt amendments to the Sacramento Area Sewer District Sewer Ordinance.
Christak will just highlight what it is that.
So this is the Sacramento Area Sewer District Sewer Ordinance. This is a 2-step process, and we're in the 2nd step now. Your last board meeting, you approved introducing the ordinance. The ordinance changes are fairly minor. They are for clarification. There aren't really any significant policy changes, anything like that. So at this point, unless you have some questions, the recommendations in the staff report, but we basically need to open a public hearing and close it and complete the other items on the recommendation.
Okay, any questions of staff? We'll open public hearing. If there anybody from the public that would make a comment or question? Close the public hearing. Is there a motion?
Move the item.
Second.
Moved by Director Peters, second by Director Bruins. All those in favor?
Aye.
Opposed? Abstained.
That's your last item.
And it looks like the rest of our items are received in files. So if there's no more comments or questions of board members, we're adjourned.